In selection, formation, callus is a mass of undifferentiated cells that can be induced from plant tissues, stems, or roots in response to growth regulators in a culture medium. Selection process. Once callus is induced, it is often subjected to a selection pressure, such as the presence of specific chemicals, for example, herbicides, antibiotics, or physical conditions, for example, light intensity, temperature. The goal is to select cells or calluses that survive or show the desired trait, resistance to a disease or chemical, while others are discarded. Some clonal variation. Tissue culture, genetic variations, called clonal variations, can occur, which may lead to potentially useful traits. Selection helps isolate and propagate these variants. For example, a may develop resistance to a pathogen through genetic mutations during tissue culture, and can help identify and grow these beneficial plants. Engineering. In some tissue culture processes, plants are genetically engineered to carry newer after transformation. For example, using agrobacterium, selection is used to identify plants that have integrated the foreign gene. A selectable marker gene, like an antibiotic resistance gene, is included. Plant cells that have taken up the new gene will survive in the presence of, while untransformed cells will be killed off. Selection of disease-resistant plants. Plant tissue culture. Selection can be applied to identify and propagate plants that show resistance to certain diseases. For example, if a plant species is susceptible to a particular pathogen, tissue culture techniques can be to isolate resistant plants through selection under controlled conditions, such as inoculating cultures with the pathogen and observing which survive. Selection for improved traits. In some cases, tissue cultures are used to select for plants with enhanced qualities such as faster growth rates, improved fruit or flower quality, higher nutritional content. The plant tissues that show the best expression of these traits can then be propagated and used for further development. Methods of selection. Antibiotic or herbicide resistance. This is a common method where a plant tissue is exposed to a specific antibiotic or herbicide, and only those cells that are resistant will grow. Environmental stress. Plants may also be subjected to abiotic stresses, like high salt concentrations or drought. Select for stress-resistant varieties. Sterilization and inoculation. Sterilization and inoculation are essential steps in plant tissue culture to ensure successful growth and contamination-free cultures. Here's a detailed explanation of each process. 1. Sterilization in plant tissue culture. Sterilization is a critical step to prevent microbial contamination, such as bacteria, fungi, and viruses, that can interfere with the growth of plant cells or tissues in culture. This ensures that only the plant material of interest grows and proliferates. Steps in sterilization. Surface sterilization of plant material. Sodium hypochlorite, bleach, usually 10 to 20% solution, for 5 to 15 minutes, depending on the tissue. Mercuric chloride, HgCl 2 0.1% solution, for a short time, 1 to 2 minutes. This is a stronger disinfectant, and it's toxic, so it must be carefully handled. Ethanol, 70%, for a few seconds to disinfect. Sterilizing tools and equipment. Autoclaving. All tools, growth media, and containers used in tissue culture should be sterilized by autoclaving at degrees Celsius, 15 pounds per square inch, for 15 to 20 minutes to kill any microorganisms. Laminar flow hood. A laminar FBW hood, sterile air environment, is used during inoculation and other manipulations to avoid airborne contamination. 2. Inoculation in plant tissue culture. Introducing sterilized plant material, such as explants, seeds, or tissues, onto the it will begin to grow and multiply. Preparation of the culture medium. A nutrient-rich medium, usually Murashige and Skog, MS, medium, is prepared to support plant growth. This medium contains essential macronutrients, nitrogen, potassium, micronutrients, iron, manganese, vitamins, sugars, usually sucrose, and plant growth regulators, for example, auxins, cytokinins. Of sterilized explants. After surface sterilization, the plant tissue, such as a leaf, stem, root, or shoot tip, is carefully placed onto the sterile culture medium under aseptic conditions, usually inside a laminar flow hood. The explants are usually transferred to petri dishes or culture jars. Sealing and incubation. After the inoculation, the culture vessels, such as petri dishes or flasks, are sealed with parafilm or plastic caps to maintain sterility. The vessels are then placed in an incubator or culture room at a controlled temperature, usually around 24-27 degrees Celsius, with a proper light cycle, typically 16 hours of light and 8 hours of darkness, depending on the plant species being cultured. Monitoring and subculturing. Over time, the explants should begin to grow and divide, forming callus, undifferentiated tissue, or shoots, roots, depending on the conditions and growth regulators in the medium. Cultures are monitored for growth. Death of tissues are promptly dealt with. Subculturing, transferring tissues from one culture medium to another, is done periodically to maintain healthy cultures and induce further growth or differentiation. Callus culture and cell suspension culture. Callus culture and cell suspension culture are two important techniques in plant tissue culture used for the propagation of plants, studying plant development, and generating plant cells for research and biotechnology applications. Here's an overview of both techniques. 1. Callus culture is an undifferentiated mass of plant cells that can be induced from various plant tissues, for example, leaves, stems, under suitable conditions in vitro, in a laboratory setting. What is callus? Rapidly dividing plant cells. It is typically induced by placing explants, small nutrient medium containing specific growth regulators, hormones, mainly auxins and Callus consists of non-specialized pieces of plant tissue. Cytokinins is an important step in regenerating whole plants via somatic embryogenesis, formation of embryos from cells, or organogenesis, formation of shoots and roots from callus cells. Of callus culture, small pieces of plant tissue, for example, leaf, stem, root, are excised from a parent plant. These explants are surface, sterilized to remove any potential microbial contaminants. Callus induction. The sterilized explants are placed on a solid nutrient medium, usually agar-based, containing appropriate concentrations of plant growth regulators. Auxins, for example, 2,4-D, Na, promote cell division and callus formation. Cytokinins, for example, BAP, kinetin, can help with cell proliferation and regeneration. Type of growth regulator combination and concentration affects whether callus formation, shoot, or root induction occurs. Subculturing. As the callus grows, it may be subcultured, transferred to a fresh medium, to promote continued growth and to initiate further differentiation. For example, shoot or root formation. Differentiation. Callus cells can be induced to differentiate into shoot, root, or even embryogenic tissues, depending on the medium and environmental conditions. Applications of callus culture. Embryogenesis, inducing callus to form somatic embryos. Regeneration of whole plants. Callus can be used as a source for the production of plantlets. Genetic transformation. Callus cultures can be used to introduce foreign genes into plant cells via techniques agrobacterium, mediated transformation or gene gun. Secondary metabolite production. Callus cultures can be used to produce valuable secondary metabolites, like alkaloids, flavonoids, or essential oils, for pharmaceutical or agricultural purposes. Suspension culture. Suspension culture is a technique where individual plant cells or small clumps of plant cells are in a liquid medium under continuous or intermittent agitation, providing a dynamic environment for cell growth and differentiation. Cell suspension culture. The callus or single cells are transferred to a liquid medium that promotes division and growth. This culture is more dynamic than solid phase callus culture because they are suspended in a liquid, often with continuous shaking to provide oxygen and nutrients. Primarily used for the production of secondary metabolites, large-scale cell propagation, or for research into plant cell biology and genetics. Process of cell suspension culture. Establishing the suspension culture, a small piece of callus or a few isolated cells are placed in a liquid nutrient medium containing essential nutrients, macro and micronutrients, sugars, typically sucrose, and growth regulators. The liquid medium is typically agitated by shaking, in a shaker or bioreactor, to ensure uniform distribution of nutrients and gases, such as oxygen, throughout the culture. Cell proliferation. The cells in the suspension culture divide and grow, forming a homogeneous cell suspension. The density of the culture is monitored and adjusted by transferring cells to fresh medium as needed. Maintenance and subculturing. The cells are periodically subcultured into fresh medium to maintain cell growth and to prevent the accumulation of metabolic waste products. For large-scale cultures, bioreactors, large vessels, may be used for the continuous cultivation of cells. Harvesting. The cells are harvested when they reach the desired density or when enough secondary metabolites have been produced. Applications of cell suspension culture. Secondary metabolite production. Suspension cultures are ideal for producing valuable compounds such as alkaloids, phenolics, terpenoids, and other bioactive metabolites. These can be used for pharmaceutical, cosmetic, agricultural applications, genetic studies. Suspension cultures allow for the study of plant cell physiology, genetics, and the effect of different growth regulators or stress conditions on cell growth. Biotransformation. Suspension cultures can be used in biotransformation processes where plant cells or enzymes used to convert substrates into desired products. Tissue culture engineering. Suspension cultures provide insights into plant cell growth kinetics and can be scaled up in bioreactors for industrial purposes. Various media preparations. MS, B5, SHPCl-2, Murashi and Skoog. MS. Medium is one of the most widely used culture media in plant tissue culture. It is a rich medium that supports the growth of a wide variety of plant tissues and is particularly effective for formation, regeneration, and plantlet growth. Nitrogen sources. Ammonium nitrate, NH4NO3, potassium nitrate, KNO3, calcium nitrate, CaNO32. Phosphates. Potassium dihydrogen phosphate, KH2PO4, and calcium phosphate, CaPO4. Magnesium sulfate, MgSO4, iron source, iron, 3, chelate, Fe, eta, as well as trace elements such as manganese, zinc, copper, molybdenum, and boron. Nicotinic acid, pyridoxine, thiamine, B1, and inositol, sucrose, as the energy source, typically 3 to 5%, agar, solidifying agent, if required. Auxins, for example, 2,4-dichlorophenoxyacetic acid, 2,4-D, Na, and cytokinins, for example, BAP, kinetin, are added depending on the purpose. Callus induction, shoot regeneration, root induction. Medium is used for general tissue culture, including micropropagation, regeneration, and even the transgenic plants. B5, Gambox B5, medium, B5, Gambox B5, medium. B5 medium, developed by Gambog and colleagues, is another common medium used in plant tissue culture. It typically used for cell suspension culture and somatic embryogenesis but can also be used for other types of culture. Composition of B5 medium, macronutrients, ammonium nitrate, NH4NO3, potassium nitrate, KNO3, calcium nitrate, CaNO32, magnesium sulfate, MgSO4, and potassium dihydrogen phosphate, KH2PO4. iron chelate, Fe, ETA, manganese sulfate, zinc sulfate, copper sulfate, boric acid, molybdenum, and calcium chloride, nicotinic acid, pyridoxine, B6, thiamine, B1, and myo-inositol. Sucrose, 3 to 5%. Growth regulators, depending on the desired effect, auxins and cytokinins may be included, agar for solidifying the medium, if needed. B5 medium is widely used in studies of plant cell culture, particularly for the production of secondary metabolite cell suspensions, and the study of plant cell physiology. It's also commonly used in plant breeding and genetic engineering. SH, Schenck and Hildebrandt, medium, SH, Schenck and Hildebrandt, medium, and Hildebrandt, SH, medium is another common medium, particularly for callus induction and the growth certain plant species. It is often used when MS or B5 media are not optimal for certain species. Composition of SH medium, nitrogen sources, for example, ammonium nitrate, potassium nitrate, calcium nitrate, magnesium sulfate, and potassium phosphate. Micronutrients. Iron, Fe, ETA, manganese sulfate, copper sulfate, zinc sulfate, and boric acid, vitamins, thiamine, B1, pyridoxine, B6, nicotinic acid, and myo-inositol. Sucrose, usually 3 to 5%. Growth regulators, auxins, for example, 2,4-D, and cytokinins, for example, BAP, are added according to the experimental goal agar, if required for solid media. PC, Powell and Chapman, medium, PC, Powell and Chapman, medium, PC medium, developed by Powell and Chapman, is another plant tissue culture medium that is generally used for the regeneration of plants, particularly those with more specific requirements for mineral nutrients. Composition of PC medium, potassium nitrate, KNO3, calcium nitrate, Ca, NO32, magnesium sulfate, MgSO4, and ammonium nitrate, NH4NO3. Micronutrients. Iron, Fe, ETA, manganese sulfate, zinc sulfate, copper sulfate, boric acid, and calcium chloride, vitamins, thiamine, B1, myo-inositol, pyridoxine, B6, and nicotinic acid. Sucrose, typically 3 to 5%. Growth regulators, depending on the specific needs of the culture. Growth regulators like auxins, for example, 2,4-D, IAA and cytokinins, for example, BAP, may be added. PC medium is particularly suitable for specific tissue culture experiments like shoot regeneration and root in some plant species. L-2 medium, L-2 medium. The L-2 medium is used primarily for root induction and is considered one of the best media for adventitious root formation. It is generally used for the in vitro rooting of plantlets that have been regenerated through tissue culture. Composition of L-2 medium, macronutrients, NH4NO3, nitrate, KNO3, calcium nitrate, Ca, NO3, 2, magnesium sulfate, MgSO4, and ammonium nitrate. Iron, Fe, ETA, manganese sulfate, boric acid, copper sulfate, and zinc sulfate, vitamins, thiamine, B1, pyridoxine, B6, myo-inositol, and nicotinic acid. Sucrose, 3 to 5%. Regulators, typically auxins like indole-3, butyric acid, IBA, or naphthalene acetic acid, Na, are included in use routing. Digital learning content. Parola University F in YouTube, www.parolauniversity.ac